Dr. Lidan You, who is the Erwin Edward Hart Professor of Mechanical Engineering and Biomedical Engineering at the University of Toronto. So Dr. Yu received her PhD from the City University of New York and her postdoc at Stanford. So she has received a lot of awards, among them the Early Research Award from the Ontario Minister of Research and Innovation. She also received the Dugan Medal from the Canadian Society of Mechanical Engineering, and is elected a Fellow of Canadian Society of Mechanical Engineering and a Fellow of American Society of Bone and Mineral Research. And so her research is focused on solving biomechanical questions in the musculoskeletal system. Mostly, she looks at them from the cellular level. She looks at the anti-resorptive effect of mechanical loading on bone tissue, looks at breast cancer, bone metastasis, prostate cancer metastasis, and looks at the mechanosensitivity, especially as it relates to osteocyte in diabetic condition, and the advanced microfluidic system for bone cell mechanotransaction study. I remember meeting Dr. Yu at the skeletal biology structure and regeneration study section of the NIH, and I said, "Oh, look, there's another Canadian." So the title of the talk is Role of Osteocytes in Regulation of Breast Cancer Bone Metastasis. Dr. Yu, please.
Thank you, Paxton, for the nice introduction. And it's really my great pleasure to have this opportunity to share our research here with colleagues in McGill. So, so first I want to start with the introduction. What is the problem we're dealing here today? So it's about the bone metastasis, and it's about a high factor risk in the metastatic breast cancer patients. So basically, 1 in every 8 women will develop invasive breast cancer in North America, and breast cancer bone metastasis is a major issue in spread of cancer to bone. 85% of the advanced breast cancer patients suffers from it, and it has the feature of osteolytic bone lesion and found in mostly stage 4 breast cancer patients, and can be fatal, can be very painful. So that's why we believe reducing fracture risk in metastatic breast cancer patients is of great clinical significance. So what is the solution then? Probably from the anti-resorptive point of view, we can do either chemical or mechanical. Chemical is already in practice. So basically using biphosphonates, we can effectively shut down osteoclast activity, prevent bone loss. And it is already a standard adjuvant treatment for metastatic breast cancer patients. However, it comes with limitation. There's patients with low adherence because of the gastrointestinal tolerability. And there's also potential side effects such as osteonecrosis of the jaw. The other option is mechanical loading or exercise. As we all know that loading is beneficial for us, and not only for our heart muscle, but also for our bone. As you can see, the bone in the playing arm of a tennis player is denser, stronger compared to the the bone in the non-playing arm. On the other side, if we're in a microgravity environment, then we will suffer severe bone loss. So then what happens to bone and cancer when we do exercise? Well, when we do exercise, our bone is for sure loaded, and there's mechanical loading on bone. And our circulatory system, right, where if we have breast cancers in so the tumor in the primary site, if some of the tumor cells through the introversion get to the circular system, and also through the extroversion, then later on get to the bone site. So of course, this process can be extremely long, and some of the cancer cell can be dormant, can stay in dormancy for years. But then our thought is that when we do exercise, we're for sure is exciting our bone cells, and then how that will affect the cancer cells, either in dormancy or in the sacral system, or even in the much earlier stage. So we wonder what's going on and what is the effect of exercise on this whole process. So we have 4 key players I want to stress here. Of course, in addition to cancer cell is the major player here. And then in the bone microenvironment, we have osteoclast, which is the bone resorption cell. We have osteoblasts, which are responsible for forming bone. We have osteocyte, which are believed to be the major mechanosensors. We also have endothelial cells, which are actually in the first line in contact with cancer cells when the cancer decides to exit and extravasate and move towards the bone surface. So there's already extensive study for many years looking at the crosstalk between osteoclast cancer cells, which shows that it actually forms a vicious cycle and they can affect each other, making the situation worse. There's also a lot of studies looking at the crosstalk between osteoblasts and the cancer cells, but not much, at least several years ago, not much on osteocyte and not much on how osteocyte could play a role here in regulated bone metastasis, in regulated cancer cells behavior. So osteocyte is the major interest in our lab, right? I've been looking at osteocyte mechanosensitivity for many years. So osteocyte very, let me just change the, arrow options. So I'll try to move. Okay, how do I? Sorry about that. I need to turn off the read. Okay. So osteocyte, this is the in vitro study looking at how osteocyte responds to fluid flow. As you can see, osteocyte under steady flow, which is 1 Pascal. The cells are rather relaxed, not doing much, but osteocyte under dynamic fluid flow shear stress, also at 1 Pascal. You can see the cells are much more dynamic, much more eager to reach out to each other with their protrusions, trying to connect, trying to crosstalk with each other. So as you can see, so they are very sensitive to mechanical loading, not only to the magnitude, but also to the type of the mechanical loading. So next, one of the earlier events of osteocyte response to mechanical loading is the calcium, intracellular calcium response, as you can see here. After you apply, in this case, it's actually pressure. So hydraulic pressure, dynamic hydraulic pressure, as you can see, the cells response with instantaneous increase of intracellular calcium concentration, shown as the pink color. And if you look at the percentage of cell response, the baseline, which is the cells under no pressure, and the cells under pressure, you can see there's a much huge increase in terms of percentage of cells responding with intracellular calcium increase. So the rationale for this study is that we know that exercise and metastasize the cancer cell both affect the bone remodeling, right? Cancer crosstalk to osteoclast, crosstalk to osteoblast. There's already extensive evidence suggesting that. But there's very limited knowledge on the role of osteocyte in regulating cancer cells. And also exercise is a common intervention strategy suggested by the doctors to cancer patients. And there's maybe we can optimize exercise program to improve the living quality while reducing risk for bone metastasis. So all this started from 10 years ago, right? 2012 summer. And that time, my major focus is on osteoporosis, on diabetes. So I actually just have recruited summer students working on this as a side project. So that time we already did some vibration studies, looking at the vibration on osteocyte mechanosensitivity. So I told the students, go ahead, just vibrate the cells, look at how that will affect the cancer cells. And 4-month study gave us result like this. So basically it shows that if you vibrate osteocyte, take the condition medium, culture cancer cells in the condition medium, you actually see increased cancer proliferation and migration, and also decreased cancer apoptosis and migration. We were really struggled with this result because we thought that supposedly exercise should be good to patients. So whatever result we have here, does that suggest that we shouldn't tell the patient to do exercise at all? But then that's just vibration. I thought maybe that's a loading specific issue. So in 2013, I recruited another summer students, undergrad students. So actually told him, why don't you share osteocyte and then take the condition medium and the culture cancer cells, take a look at what happened. And exactly the same result. So that got me thinking, right? So what's going on here? And then I go back to the picture I showed you earlier, the major player here. So I realized that in the bone marrow environment, osteocyte is not the only cell, although it's the major cells, but it's not the only cells plays a role here. So there's osteoclast, there's endothelial cells, there's osteoblasts, right? So in 2014, we pretty much done with the diabetes project and other projects. So I recruited a graduate student and started to work on this. So Vivian Yuhengma joined us, and she started this project dedicated to looking at osteocytes' role in regulating bone metastasis. So basically, what she did is she applied free flow, and of course, there's a control with osteocyte in no flow, and then take the condition medium, and then culture osteoclast and endothelial cells here. So basically, looking at osteocyte crosstalk to cancer cells via a third player, either osteoclast or endothelial cells. We put these two step cells in between and look at how that will affect the cancer cells' fate. And we look at the migration and apoptosis. What we found is very encouraging, and we found that flow on osteocyte, right? And if we take that condition medium, culture osteoclast, and then take condition medium of that, culture cancer cells, we found dramatic decrease in terms of cancer cell migration. So as shown here, and we use MCF7 as a control. And also we show that flow on osteocyte and osteoclast can actually promote cancer cell apoptosis, shows in this figure. So we next looking at actualization. So basically, in this case, we use transwall system, and we culture endothelial cells on the transwall, and then we have culture osteocyte at the bottom and the cancer cell at the upper chamber, and we look at whether the flow versus no flow can actually affect the cancer cell actualization. And turns out yes. The answer is yes. So flow on osteocyte can actually downregulate actualization of cancer cells via endothelial cells regulation. And we also look at whether through the endothelial signaling, whether that will affect the cancer cells apoptosis. And yes, the answer is yes. And again, we see the increase in cancer cell apoptosis when you treat the cancer cells with the double condition medium from endothelial cells cultured with flowed osteocyte condition medium. So in summary, for this product, what we found is that if you simply do the co-culture of osteocyte and breast cancer cells, you actually see loading can promote cancer. However, if you put osteoclast and endothelial in between, so osteocyte can regulate osteoclast and endothelial cells, and then later on that will result inhibition of cancer in terms of downregulation of migration, downregulation of transendothelial migration, and increase of upregulation of apoptosis. So then we ask whether the signaling can actually also change the cancer cells gene expression. So the idea is that endothelial cells, right, the permeability might also be changed, and then the adhesion might be changed. All this can actually affect the fate of cancer cells actualization. And we also want to know whether the endothelial cells can affect the cancer cells gene expression in terms of the potential of a metastasis. So what we did in the next project, again, this is Vivian's project, she again flowed osteocyte and then take the condition medium and culture endothelial cells, and then test the permeability using the tracer, and then also see the cancer cells on the endothelial cells, and then do the flowing later to see the adhesion, whether the adhesion of the cancer cell to endothelial cells is changed. And then later on, we also did an RNA-seq looking at the cancer cells gene expression. So what she found is flow on osteocyte through endothelial cell regulation can actually decrease flow on osteocyte can actually decrease endothelial permeability and can also decrease adhesion of cancer cells to endothelial cells. And from the RNA-seq study, which is we did RNA-seq on the cancer cells, right? The cancer cells is treated with condition medium from endothelial cells, which is treated with the condition medium from either flow or no flow osteocyte. And we found basically 35 gene ontology terms downregulated, and basically 5 genes downregulated in the cancer pathway. And specifically, maybe I should skip this. We're looking at MMP9 and FISOD4. These are 2 downregulated in our RNA-seq result. And MMP9 is the collagenase, and it shows, there's evidence shows that it's correlated with bone metastasis. And FISOD4 also is a receptor that transduces Wnt signals. So we decided to confirm or validate our RNA-seq result by doing a qPCR, and what we found is consistent with our RNA-seq result. So osteocyte, flow, no flow, and culture endothelial cell, and then culture cancer cells downregulate MMP9 expression, and also downregulate FISOD4. And we also block MMP9 inhibitor, which show that basically that we can shut down or we can blunt the invasion caused by the MMP9. So in summary, we show that loading on osteocyte can decrease permeability of endothelium, and then can also decrease adhesion by breast cancer cells. And also through the MMP9 FISOD4 downregulation, we can actually affect bone metastatic breast cancer cells in terms of downregulation is invasion. Right. So that's what have been done before 2017. So it's a 3-year project. We got what we expected. We're really excited. But then we realized that there's a huge limitation with this approach, with this condition medium approach. There's no real-time communication. There's no real-time capability of monitoring of migration, and the communication is unidirectional. There's no bidirectional communication. Cancer cells should also be able to affect osteocyte. So we decide to take another approach to develop a microfluidic platform to study this. At that time, we were actually looking at existing platforms available. So there's a lot of organ on chip concepts already there, and people are talking about bone on chip, bone metastasis on chip, cancer on chip. So we invested all the chips available at that time, and even now things doesn't change much. So basically, many of the chip doesn't have the endothelium structure there, and none of the system offer the bone free flow, which we believe is critical for osteocyte metabolism and function. So we decide to then develop our own platform. So the students for this project aims to develop a physiological relevant mechanical stimuli platform for osteocyte, and then determine whether the effects of mechanical loading from physical activity can on breast cancer metastasis on osteocyte regulation is indeed the case. So basically validate the conditioning study result. So Christina Schöting-May is the student's work on this project. So she developed this microfluid project, microfluid platform. Basically, there's two chambers. Maybe this one is better. Okay, so basically we have a system can host 6 microfluid system. In each system, it has these 2 microfluid channels. And if you look at the 2D cross-sectional view, on the left-hand side is the channel where osteocyte can be seated and experiencing mechanical challenging. And then on the right-hand side, we can form artificial blood vessel by seeding endothelial cells form this circular channel. And then we can fill in, we can flush in the cancer cells. And then there's side channels cross to connect these 2 major microfluid channels, allowing the cancer cells can get across to do the migration and the transendothelial migration. So this is the dimensions of our design of the system. So basically the distance we put it, this distance between 2 chambers is 300 micrometer. We believe that's close to what's going on in the in vivo situation, where the distance usually between osteocyte and bone surface cells or cancer cells is in the 100 micrometer scale. And so the outcome variables to be assessed is the breast cancer actualization distance and also the percentage of actualization side channels. So first we validate, we have a nicely formed endothelium layer, endothelial cell layer line along the channels. And we have MDMB231 cancer cells injected into the chamber. We have HUVEX here, and then we can look at the migration of cancer cells across the channel. Remember, on the left-hand side is the chamber host of Y4 cells, which are the osteocyte cell line. And this is basically, this is the osteocyte without flow. So, and this is osteocyte underflow. You can see that under oscillatory free flow, there's not much migration going on, but when the osteocyte is under static condition, you can see that there are actually a lot of migration of cancer cells towards the osteocyte channel. So we found that with oscillatory free flow, the actualization distance is decreased. The actualization percentage, this is distance, this is percentage, is also decreased dramatically. And we also found, then the next question is, whether this result is actually due to the flow itself, because when you have flow versus no flow, the pressure will be different. Maybe it's not osteocyte releasing anything. Maybe it's just the endothelial cells and the cancer cells sensing the pressure and automatically making the adjustment and made the decision to either migrate or not, right? So to check on that, what we did is we removed the osteocyte from osteocyte chamber. So basically on the left chamber, we didn't see any cells. So it's an empty chamber, but then we put a flow versus no flow, right? So that's what this experiment is about. So on the left-hand side is static, on the right-hand is flow. And we did a paired comparison, paired static test, paired t-test, and we found there's no difference between the static versus flow group, both in terms of distance and percentage. So we're sure that whatever we observed in the previous slides is actually indeed due to the signalings released from osteocyte under mechanical loading. So in conclusion, we enabled, in this part, physiologically relevant bone fluid stimulation and real-time intercell signaling between the 3 different cell types. And we confirmed the double condition medium study, which is mechanically stimulated osteocyte can actually regulate transendothelial breast cancer actualization. So this study is featured in Science Translational Medicine, and I like the title that the editor or the author come up with, which is a workout plan to provide metastasis. So then the question is, come back to the exercise problem, to exercise or not, right? Obviously, what we showed, the evidence showed here is that exercise is good, right? Exercise loading on bone can indirectly signaling to cancer cells and provide cancer cells migration. So seems everything is good. However, this issue here, oh, my apologies for the typo. So it should be low. So it's an issue which is low adherence due to the following reasons. For some people with overweight or obese situation, and some patients with physical fatigue, mental stress, or has the fear for unintended injury. So that all cause a low adherence to the exercise. So then safe, effective exercise alternative is actually needed. So then we think about the vibration, right? So the vibration, there's a lot of investigation on this topic already. By that time, it shows that vibration can increase bone mass, benefit muscle strength, good for balance. There's many benefits there. So we think maybe vibration can serve as alternative. And from our lab, actually a long time ago by another student, so Esther Lausch, so she published this in 2010, showed that vibration can actually increase COX-2 gene expression in osteocyte and also downregulate RANCL, upregulate OPG, shows that vibration has the osteogenic effect and also anti-resorptive effect on osteocytes. So then Susie joined us in 2020. She can't confirm on this, but what she does, she used our already developed microfluidic chips and then put the osteocyte on the chips and also cancer cells and the endothelial cells on the chips. The system we use exactly the same as I just showed you. But instead of doing the fluid flow, she actually vibrated the platform. And then looking at, and also she put in hyaluronic acid, which is a biphosphonate at 5 micromolar for 3 days. So what she shows is that vibration and in the presence of biphosphonate can actually downregulate activation of cancer cells in terms of distance. So all this supporting the idea that vibration is beneficial and can serve as a exercise alternative. However, this is a big however. The whole body vibration or the vibration seems to work on young patients only, not effective for old patients. This comes from 2 clinical trials and 1 animal studies. All shows consistently that if you do vibration to young people or young patients or young animals, effective, but doesn't work to old patients, to old animals. So seems like enhancement is required for vibration. If vibration is the answer, then we need something extra to enhance it so that vibration can be effective for old patients. So we're thinking about potential strategy to enhance vibration effect. First, the potential cause for the losing effectiveness of vibration in old patients. We think maybe it's because of the low osteocyte mechanosensitivity. That has already been demonstrated. So then the potential target could be osteocyte itself, and it could be whether we can come up with a way to improve osteocyte mechanosensitivity to vibration. So talking about mechanosensitivity of osteocyte, as I showed you earlier, one of the early response in osteocyte response to mechanical loading is calcium, intracellular calcium concentration. So calcium can come from outside, right? So from extracellular environment. Now through the ion channel, piezo1 is one important ion channel. It's actually a mechanosensitive ion channel. So piezo1 is expressed in osteocyte. and it's actually highly expressed in osteocyte, as you can see in this figure. So the idea is that if we can take advantage of what we know about PSO1, if we can use some chemicals to actually activate PSO1 or to lower the threshold of opening PSO1, then maybe we can help all the patients by improve osteocyte mechanosensitivity. So the rescuer here is the UDA1. So UDA1 is a small chemical, it's a small molecule, it's actually a PSO1 activator. So it can bind to the PSO1 channel protein, and then can actually lower the threshold of opening of the PSO1 in response to mechanical loading. So we think maybe we can take advantage of what we know about the property of this small molecule UDA1, and then use UDA1 plus vibration to activate osteocyte. So my collaborator, Dr. Lidan You from University of Delaware, so they did a proof of concept study looking at, so this is an in situ study. Basically what they do is they have a bone isolated from animal, and then they do the loading. So and then they look at the osteocyte embedded in the matrix. They look at osteocyte calcium response. So what they show is that without no UDA1 treatment under 8 Newton loading, right, you see some oscillation of calcium. No loading with 10 micromolar UDA1, you see this oh, I don't, okay, so it should be, okay, so basically this is the number of normalized to, I forgot what is this. This is so this should be this should be the figure. So shows the no. Oh, this is no loading. My apologies. This is no loading plus 10 micromolar UDA1. So you see not much calcium response, and this is loading same magnitude, but with 10 micromolar UDA1, you see this huge increase in the calcium concentration, demonstrate that UDA1 can enhance the loading's effect. So then, so we also collaborate with Dr. Fei Geng from McMaster University. So Dr. Fei Geng is a biologist, has expertise on YALP signaling. So what we did is we vibrate our osteocyte, and then we look at the staining of YALP. And then we also did some nuclear translocation study. And we sent it ourselves to their lab, and then they did imaging and analysis. And what we found is that with UDA1 and vibration, basically the UDA1 enhanced the vibration. So HFLM is in the high frequency, low magnitude vibration, can actually activate the YALP nuclear entry in osteocyte, so has the strongest effect in terms of YALP translocation. And we all know that YALP is an important mechano transduction transcription factor, is the key player in terms of regulation of many downstream bone formation and bone resorption bone markers, gene expression. So then we wonder whether whatever we observe through vibration and the UDA1 through vibration is indeed caused by through PSO1 activation. So then we collaborate with Dr. Jin-Xiong Hu from University of Arkansas for medical sciences. So their expertise is on PSO1 channel, and they do a lot of siRNA, shRNA, animal knockout, knockdown model. So we learned from them and then did a silencing of PSO1 through using siRNA and shRNA. And what we found is that if we shut down the PSO1 and what we observe in terms of response, here we're looking at relative mRNA level in PSO1. So we look at two genes, PSO1 gene and COX2 gene. So without silencing, we can see vibration upregulate piezo1, vibration upregulate COX2. However, if we knock down piezo1, this is actually a result from siRNA. So if we knock down piezo1, the response is dramatically decreased. So basically demonstrate that what we observe of osteocyte in response to mechanical loading, in this case vibration, is indeed mainly caused by the piezo1 channel activation. So then we look at whether the UDA1 treatment can indeed enhance the vibration effect on two things. One is resorption, the other is cancer. So resorption, we look at osteoclast formation. Cancer, we look at cancer migration. So the left-hand side figure, we look at osteoclastogenesis assay. So what we did is we look at the number of nuclear per cells. We quantify the we quantify the osteoclast, and we group them by the number of nucleus per cells. So we found that looking at control, which is no treatment, and then UDA1 only without vibration, or vibration only, which is in blue, and or vibration plus UDA1, which is in green. As you can see, for the smaller osteoclast, there's not much difference. However, for large osteoclast with nucleus numbers larger than 10, we see significant decrease in the number of raw osteoclasts. So we also look at a cancer migration assay. We observed a similar effect. Basically, the UDA1 indeed enhanced the vibration effect by further inhibition of cancer migration. So then we look at whether we can repeat this and have this observe the similar effect on chips. So we did UDA1 and plus vibration on chips, and then we look at a cancer cell activation. So what we found is that, so we have two time point, day 2 and day 4. Again, this is on chip with osteocyte and the endothelial cells and the cancer cells seated on this chamber, on the chips. And then we basically let it grow for 2 days and 4 days. And we have 4 groups, either DMSO control group or vibration only group, UDA1 only group, or combined group. Right. So what we found is that both on day 2 and day 4, we see the decrease of actualization distance in the combined group. However, UDA1 only seems also has very strong effect. So compared to the control group, vibration only group, vibration only also have strong effect compared to the control group. So then the next question, so far we basically showed evidence suggesting that UDA1 is indeed a good helper in terms of enhance the vibration effect. And the next question is UDA1 safe to use, right? Is that toxic? So we did live diet staining looking at osteocyte and MDA-MB231 cells, and we didn't see any difference between the variability looking at 4 different dosages. And we did a MTT assay and scratch assay for cancer cells, looking at proliferation and migration. What we found is that the UDA1 actually inhibit cancer cell proliferation and migration, even at a low concentration at 1 micromolar. And then we asked whether the UDA1 has any negative effect or any effect on osteoclast. So what we did is we tried different dosage as well, UDA1 from 1 micromolar to 10 micromolar. And what we found is for large osteoclast, large size osteoclast, we show significant decrease in the osteoclast formation. So basically the message is that yes, there is effect of UDA1 on osteoclast, but maybe the effect is beneficial because it's actually shutting down the osteoclast activity or formation. So then the next question, all we have collected so far is in vitro. Is that what happened in vivo, right? Any in vivo evidence? So again, so my collaborator, Dr. Lidan You from University of Delaware, they developed this synergetic murine model. Basically, they can do the they can inject the tumors into the tibia, right? And then they can do micro CT of bone lesions, and then looking at the weekly scans. So what they did is they inject the B6 female mice with PYA119. So this way we can actually have the immune system still working. So in this case, we believe, because we believe immune response is also one important factor in the equation. So basically, their lab did the injection, and they inject the tumor cells in both tibia, but at a very low, not dosage, but a low number. So it's only 1,000 cells per tibia. And this is what they found, right? So they found that, so two parts. One, so on the left-hand side, this is 9 weeks mice, right? But the regular mice, right? So normal mice. On the right-hand side, there's also 9 weeks, but it's actually obese mice. So what they found is that for the bone formation, this is actually without tumor, right? And you have UDA1 plus vibration, and it shows that UDA1 enhanced vibration, or UDA1 combined with vibration, can promote the bone formation, which is the pink color shows here. So the left panel here shows the periosteal mineral surface, and the right panel shows the increase in the bone formation rate in the periosteal. So both bone formation rate and mineral surface shows increase in the combined group. And for the bone with tumor loading, right? And it shows that the UDA1 enhanced vibration can actually increase percentage of the tibia free of perforation, right? So this is a tibia so it shows the percentage of tibia free of perforation. So you can see it's dramatically increased that for the vibration enhanced group. And if you look at the tibia polar moment, shows on the right panel here. So the polar moment inertia, if without enhancement, without the vibration, so it's vibration without the UDA1 and the verse vibration with UDA1, you can see the UDA1 significantly increased the tibia polar moment inertia. So both this evidence, the in vivo evidence and in vitro evidence, suggests that indeed osteocyte plays an important role in mechanical loading regulated breast cancer bone metastasis. And UDA1 enhanced vibrations can actually have an additive effect on prevention of bone loss and inhibition of cancer cell activation. So where are we now, right? Basically, I already presented the conclusion slide, supposed to be the last slides of my presentation. But I feel like maybe it's good to have an overview of what's going on in this specific field, right? We're talking about osteocyte and breast cancer bone metastasis. So this is actually indeed the slides I spend most of the time making it. So this is the basically I want to show which groups are doing what in this small field for now, right? So Dr. Maureen Lynch from University of Colorado, her group is one of the pioneers looking at loading, how loading affects tumor burden in bone, right? And they published a beautiful paper in GBMR looking at injection of cancer cell into the tibia, and then they do the loading, shows that the loading can significantly decrease or inhibit tumor, decrease the bone tumor load burden. So then recently they've been heavily invested their efforts on looking at osteocytes mechanosensitive change in the presence of cancer cell. So I've been talking about how osteocytes regulate cancer. Their focus is really looking at how cancer affects osteocyte mechanosensitivity. So that's their contribution. And then, of course, me and Professor Lidan You from University of Delaware, we started our collaboration a long time ago, but in terms of this specific topic, we started about 5 years ago. I'm looking at in vitro side mechanism study. She's looking at in vivo side how vibration can actually indeed affect mice bones, tumor burden prevention, cancer metastasis prevention, and also how vibration can potentially protect the bone lesion. So our major goals here is to aim to help old patients. We look at osteocyte inhibition of cancer cells. And then Dr. Jin Jiang from University of Texas at San Antonio. So she's also a pioneer in looking at osteocyte cancer cell crosstalk, but totally from a totally different angle. So she's looking at connexin 43, looking at the gap junctions role. So her study reveals that connexin 43 is an important player in terms of gap junction signaling, in mediating osteocyte regulation of cancer cells without loading, with loading, either way. And then Dr. Ting Tang from Shanghai Jiaotong University. So what their focus is also on osteocyte cancer, but also again from a very different or a very interesting angle. They're looking at treating osteocyte as target and using nanoparticles to manipulate osteocyte and then see how that will regulate cancer cells in terms of metastasis. And then recently, starting about 5 years ago, there's two scientists from Ireland, almost at the same time. So Dr. Stephen Verbruggen and Dr. David Hoey. So they're not from the same university. I believe Dr. Stephen Verbruggen, if he hasn't moved yet, he's from the University of Sheffield. Dr. David Hugh, he's from Dublin, so they're both from Ireland. So they have a similar result, which is very interesting. They both published paper on this, one in 2021, one in 2020. So actually, they both show that osteocyte underloading can actually promote cancer in terms of proliferation, in terms of inhibition for apoptosis, in terms of promoting migration. So this is actually something I have concern with, but I have a different opinion with, right? Because we initially also showed that, but I believe that it shouldn't just involve osteocyte cancer cell direct crosstalk. You should have other cells in between to play a role to do the intermediate as an intermediate player. So. But anyhow, I think that's still very important insight, shows that if you simply put osteocyte and cancer cell in direct contact, and when you have an exercise, what's going on? And the difference between Dr. Stephen Verbruggen group and Dr. David Hughes group is that Dr. David Hughes group focuses on the CLC XL12 signaling pathway mechanism, and Dr. Stephen Verbruggen looking at more on the organ-on-chip system of using that system to look at osteocyte cancer cell crosstalk. So I think that's conclude my presentation, and I want to thank to my students. They did a beautiful work in the past 10 years. So all the results I show are from their hard work. I want to thank my collaborator, Dr. Lidan You, looking at in vivo model, and Dr. Fei Geng looking at a YAP signaling pathway, and Dr. Jinhu Xiong provide help on the PS1 signaling pathway. Thank you very much, and I'm open to questions.
So thank you very much, Professor Yu, for that excellent presentation. Questions, please just unmute and as I think we have Dr. Gerard. Can you please ask?
I did not have a question. I was just applauding.
Okay. Yes, Derek.
Hi, Lidan. Nice to see you again.
Hi, Derek.
A very
Nice to
nice
see
talk.
you.
Very
you.
interesting. I have a couple of questions. Have you ever done some of this fluid flow using more complex 3D cultures to simulate the matrix environment?
No, that'll be really interesting to do. But for now, this is kind of our dream. We want to develop a 3D environment at the same time kind of flush free flow in the matrix. And then somehow, but because we want to make sure that the free flow shear stress is kind of like a unit, so we want to have a good control on the flow profile. So that becomes a challenge and prevent us from, I guess, develop such a structure. I wish someone can do this and we can just grab the platform and doing it. For us, we haven't been able to do it.
Yeah, there's a lot of advances in the biofabrication techniques to make fluid channels through a soft matrix. So that's somewhere interesting you might want to look. And then I had another.
I don't know. I'm aware of those technique or development, but the hardness of the matrix is always an issue. If it's in a soft matrix, the osteocytes are very sensitive to the stiffness of the substrate. So I don't think I saw there's many groups, including a Japanese group, and they developed this long time ago osteocyte culture in this collagen matrix, and looking at they did the poking of osteocyte. I'm not sure which is more representative, because my feeling is that it's just so soft. It's hard to be able to say it's a representative.
Right. One more question is, do you ever also, you apply fluid flow in a lot of your studies, and then I saw one ex vivo study where you're applying some mechanical loading. Have you ever done, you also said you did hydrostatic loading as well. Do you ever apply mechanical stretch to the cells or mechanical compression of some sort to the cells directly?
Right, that's an excellent question. So all this fluid flow business actually comes from the thought that the paradox basically is that when the bone underloading, the bone matrix, because the bone matrix is very stiff, the deformation, we're talking about 3,000 microstring or 0.03%, which is very small. If you do that type of stretch or compression to bone cells, nothing will happen, right? So then if you look at what's going on cellular level, we found that because of the paracellular matrix, because of the pressure, the loading caused the pressure gradient will drive interstitial fluid flow. The flow itself actually can serve as a major stimuli to the cells. So that's why we decided to focus on the fluid flow shear stress instead of looking at the stretch. But in 1998, Chris Jacobs et al., they actually did comparison stretch versus shear stress. What they did is they do different level of stretch and they do different level of shear stress. No, actually the shear stress is only one level. I believe it's 10 dynes per centimeter square. But stress, they did a different level. They compared. They found that only 10% above can activate the bone cells, including both osteoblasts and osteocytes. My apologies. So, but if you go lower than that, down to 3%, 1%, nothing will happen.
Okay, thank you.
Thanks.
I will have a question.
Okay.
Thank you. Thank you, Dr. Yu, for your very interesting talk. So I understand that your research is focused on breast cancer, but do you think that another solid tumor that has a tendency to home to bone for metastasis, like prostate cancer, for example, would have the same? You would have the same findings with this type of cancer?
Absolutely. I actually have another student, Kimberly Seaman. So she joined us 2 years ago. She's working on the prostate cancer side. It just given the limited time we have, I didn't put it in the slides, but we have very similar result. But what we found in the prostate cancer is that osteoblast instead of osteoclast plays a major role.
Okay. So it's due probably to the type of cancer cell that interacts with the different type of cells in the bone.
Right.
Thank you.
Thanks.
Bilal Tarabi.
Hello, thank you for the very interesting talk. Actually, my question was, is there any study that look into the effect of radiation on the activity of osteoclast and osteocytes, since, as we know, most of the patients with bone metastasis will be irradiated? Thank you.
That's a really good question. Wonderful question. We actually designed a poster in this year ASBMR looking at radiation. Actually, not this year. So this year we presented Dr. Robinson's effect and the vibration and the placebo one. So radiation for sure. So it's actually my collaborator, Dr. Lidan You, is doing that using the radiation and looking at whether the osteocytes. So they actually already show that osteocyte definitely in terms of apoptosis, huge increase. The cell are pretty severely damaged. And they show that mechanical loading, in this case, not vibration, they do this 4 Newton loading, tibial loadings. And then they show that, I'm not sure about, I think the frequency either 4 Hertz or 3 Hertz. I'm not sure about that. But they show that with the loading, the apoptosis rate of osteocyte is actually like a decreased. Yeah,
Thank
some
you.
rescue, but I don't know whether that's going to be enough.
Okay, thank you. And do we have any studies comparing the remodeling capacity of bone with cancer versus normal bone in this particular case, the effect of osteocyte and the whole process of remodeling of the creation of new bone?
To the best of my knowledge, no, I don't think so. All this osteocyte cancer crosstalk starting about 10 years ago. We're one of the first few groups started doing this, and it's actually developed very quickly. I see like 3 years ago, I attended ASBMR, and then because of pandemic, I didn't go to ASBMR. And this year, I felt a huge change. Because a few years ago when we were there, we were pretty much the only one of the 2 groups doing this. But this year, even though they didn't, I saw many groups, even though they haven't published any data yet in terms of journal publication. But I see many people looking into this. So I expect a lot of exciting results will appear very soon.
Great, thank you so much.
Yeah.
Lidan, great presentation.
Thanks.
As you were talking, I kept thinking, to exercise or not to exercise? That's the question.
Right.
So, but I'm glad that you are able to include other cell types. And so what do you think then is happening to the other osteoblast and osteoclast?
So there's many study also shows that osteoblasts are also mechanosensitive, and there's also study shows that when you do the loading and with the presence of radiotherapy, osteoblasts can also be rescued because of the under the loading. So I do think that osteoblasts, osteoclast, they all play critical role here. But I think in terms of mechanosensing, we compare the osteoblasts and osteocyte, we still found the osteocyte are the most mechanosensitive cells in terms of the magnitude of response. However, the other factor we believe that osteocyte plays a more role in terms of sensing the mechanical loading is that it's just purely by the number. If you look at the number of osteocytes in the bone matrix, right, they're in the perfect position with the huge number there. And if you're looking at the signaling, cytokine signaling, it has to come from osteocyte. I think most of it has to come from osteocyte. Osteoblasts, they're on the bone surface, but in small numbers. So that's just my thought. But I'm saying they're still very important in terms of, especially in the prostate cancer, we found that without osteoblasts, the things are also totally changed. So you have to have osteoblasts there as the direct contact with the cancer cells, prostate cancer cells.
Right. Thank you very much. So as you were transferring the conditioned media, I was thinking, are there some factors there that are being released? What are your thoughts?
Right. So we did some screening. So basically, rank OPG. So on the breast cancer cells, we're looking at a rank OPG and PGE2. So, but we definitely want to looking at a more signals. So that's why we're in collaboration with Dr. Fagan. So they have this facility. So basically, we want to do a protein screening to see what's going on in the secreted in the conditioned medium.
Okay. So my last question is, I like the way that you did your models and the use of Yoda-1. So one thing, what sort of, because in some studies they show that Yoda-1 can be dependent on the concentration used, especially for neurons. So what sort of concentrations were you using?
Right, that's an excellent question. So Yoda-1, the concentration we tried 1 micromolar to 10 micromolar. 10 micromolar is really overkill, right? So basically, if we're talking about in vivo study, we use, let me just double check, make sure I have the number correct. So we have, 1 second. I believe it's 5 milligram per kilogram, 5 milligram per kilogram.
Okay.
Yeah. So then we did a by weight, all right? So we did presentation. So we did some estimation. With that concentration we used, we did an IP injection of this Yoda-1 to mice. So that is equivalent to about 0.1 micromolar. So 0.1 micromolar concentration. So what we used in vitro, so we used from 1 micromolar to 10 micromolar. It's definitely higher than the in vivo. But even in this such a high concentration, we see that it's not very much toxic to bone cells, to osteocyte. We actually test this on the primary osteocyte. We isolated from both young mice and old mice. Young mice is more tolerant to 1 micromolar Yoda-1, but old mice, the osteocyte, we isolate from old mice. We're talking about 18 months old mice. It's actually less tolerant.
Okay, thank you very much. Any other questions?
Sure, I have one more question. I have one question, if you don't mind. Yeah, great work, Dr. Yu, and I really like the flow chambers you developed. Quite amazing, actually. So in a lot of that work, you mentioned that the Yoda-1 and activation of piezo-1 channel induces calcium oscillations, and if I'm not mistaken, it's those calcium oscillations that right. That affect the cell migration, the cancer cell migration from the with the osteocytes, right?
Right.
So I was wondering if there's other ways, if it's really the calcium oscillation, if this is the main mechanism, are there any calcium mobilization agonists and activators that could be used that would have a similar impact on the effect of cancer migration due to osteocytes?
Yeah, that's excellent question. We've been also wondering this as well. So I'm not the expert of PSO1, but so that's why we collaborate with Dr. Jin Hu Xun. So all their work is about the PSO1. So what they found is that yes, there are other ion channel, calcium channels on osteocyte. However, if you knock down PSO1, what happened to the mice is that basically they lose bone formation significantly. So this is conditional knockout, DMP1 Cre. So it's targeted to osteocyte. So basically that suggests that yes, even though there's other ion channel, other calcium channels for osteocyte, but the PSO1 is still the major one, I guess, in terms of sensing the mechanical loading.
Thank you.
Yeah, they also treat mice with UDA1. They see significant increase of bone formation. And this is actually confirmed by another group from Harvard University, Dr. Yingzi Yang group. So they show the PSO1. They also did I forgot whether it's conditional knockout. It has to be conditional knockout. So targeting osteocyte, they showed a similar effect.
Very interesting. Thank you.
Yeah.
So thank you very much, Dr. Yu. If there are no other questions, I would like to thank everyone for attending this meeting, and wonderful work, Dr. Yu. I look forward to future work. Really wonderful, excellent. Thank you very much, everyone.
Thank you all. Bye.
Thank you. Bye.
Bye.